we present a method to generate human-induced pluripotent stem cell-derived tumor antigen-specific CD8 alpha-beta single-positive T cells by using OP9 delta 1 co-culture system. This method is a powerful tool for in vitro T cell generation and will facilitate the development of in vitro-derived T cells for use in regenerative medicine and cell-based therapies. It can also be adapted for the generation of other lymphocytes like NK cells. When performing this technique, it is important to pre-evaluate the load of FBS and passes OP9 delta 1 cells consistently when cell-to-cell cytoplasmic contact begins to prevent cell differentiation and senescence. Demonstrating the procedure will be Dr. Meghan, a surgical oncology fellow from my team.
Start by checking human iPSC colonies in a stereo microscope and remove any areas of differentiation from the culture with the plastic edge of a 200 microliter pipette tip. When the colonies reach 0.8 to 1.2 millimeters in diameter, passage the human iPSCs. Aspirate the spent media and add 10 milliliters of human iPSC media supplemented with 10 micromolar ROCK inhibitor. Roll a disposable cell passaging tool across the entire dish in one direction, making sure to maintain uniform pressure and that the entire roller blade touches the culture dish. Rotate the culture dish 90 degrees and repeat the rolling. Visually confirm the proper cutting of the colonies with a microscope. They should appear checkered. Then, detach the cut colonies by gentle mechanical flushing with a 200 microliter pipette. Visually estimate